I've been asked to talk a little bit about what's new in cystic fibrosis with particular reference to some of the grant programs that we've been funded through LifeArc and through their involvement with the CF Trust, and give you a little bit of a feel for how much has changed in such a short period of time. But the big take home is going to be we aren't done yet. There's still an awful lot more that needs to be done. So I've called this New Horizons, New Challenges, and I hope you'll see why as we go through. I've got a few relationships working with various companies in this space, and I'm also the president of the European CF Society at the moment, so very much on a sort of international outlook rather than just purely UK. Those of you who don't work in cystic fibrosis probably remember from biology days at school, it's a recessively inherited condition which is multi-organ in its manifestations. We tend to think of it as a respiratory disease, but there are major digestive issues as well, also affecting the liver, the upper respiratory tract, and the male urogenital system. We now understand that CF isn't as black and white as it was when I was training as a medical student, and there are a mixture of various CFTR-related disorders, which we now recognize from people who might carry slightly milder variants of the gene mutations. And then people with CF also acquire a number of different problems related to largely actually some of the way in which we have to treat them. So some of the drugs that we're giving, for example, there are some hearing problems associated with recurrent use of aminoglycosides, same kidney problems related to that, and osteoporosis is also something that comes on with age. I'm not going to summarize the last few decades, but just to tell you where we are today, things have changed enormously since when I was training. We have a really good understanding of the basic pathophysiology of the disease, and that has led us to better understand genetics, diagnostics, and the way we treat these patients. In the UK and in many parts of the world, we have newborn screening now, which detects cystic fibrosis. So most new diagnoses are happening at about 3 weeks of age, which give us a really unique opportunity to get in and treat early. And we also have evidence-based treatments for many, many of the downstream consequences. So for example, lung infections, that's been a big topic that we've discussed this morning and will come up again a little bit later. But these treatments are very, very burdensome. And people can spend even like 2 hours a day, even when they're well, just keeping on top of their health. The really big game changer has been the arrival of these new drugs called CFTR modulator drugs. And these are oral agents which reach all parts of the body and therefore have the potential to improve CFTR function, not only in the lungs, but also perhaps in the digestive tract and in the sweat gland, which is where we do our diagnostic test. So we've got a nice, easy biomarker readout of their efficacy. The ones which have been most successful are combinations of 2 or 3 different types of molecules. And these molecules work by improving the function of the CFTR protein that the body makes faulty. So in the small proportion of people with cystic fibrosis who don't make any protein at all, that's about 5 or 10% of people worldwide, these drugs do not work. So when you hear of people who aren't suitable for these modulators, it's usually because their genetic variant doesn't produce any of the protein. So just as a little diagram then, we get the production of CFTR from translation through to post-translational maturation in the intracellular organelles. One of the commonest defects in CFTR is that it doesn't fold up properly and it gets degraded into the proteasome instead of traveling up to the cell surface where it needs to function. So what these drugs do, the modulators, correctors, will stabilize the way the protein is folded and allow it to go up to the cell surface. And potentiators are the other class of molecule which sort of kick the protein into overdrive. The CFTR protein is an ion channel and it is an active ion channel which opens and closes. So the combination of these two first get it up to the cell surface and then make it open more frequently. And the sorts of benefits that people have reaped from these drugs are really quite impressive, more impressive than anything we've ever seen with other drugs. This is just the sweat chloride concentration, so that diagnostic test I mentioned to you. People with cystic fibrosis have got very high sweat chlorides. People on placebo in this phase 3 trial maintained those high levels, whereas very quickly you see that coming down as a readout of restoration of CFTR function in the people who were on this triple combination drug. That doesn't matter too much to patients, but it's very handy for us. What does matter to patients is their lung health. And we can see here at the top the primary readout of lung health in obstructive lung diseases, which is called FEV1. You can see that goes up very, very quickly and then is maintained for that 6-month period of this trial. And also that really relates very much to the frequency with which people need to get admitted to hospital because they have flare-ups called exacerbations. And you can see here quite a big hit on the frequency of those exacerbations. And this is really important because every time somebody with CF has an exacerbation, there is potential for them to permanently lose some of their lung function, which then never comes back. The great thing about this data set is it is on a group of patients who have got one CFTR variant that can respond to these drugs and another one that can't. So you can see how much can be achieved just by giving them a sort of heterozygote level of restoration. So these drugs are suitable for people with one or two of these particular common F508del variants. But they're not only suitable for those people. Although they were designed initially for those people, they actually work for a huge number of different types of CFTR variants. There are over 2,000 different variants that have been described in CFTR in various parts of the world. By far the commonest is that F508del, but there's lots of others as well. This is just a snapshot of what the modulator landscape looks like at the moment in terms of licensed indications. And you can see that for one of these, for a single agent ivacaftor potentiator, which is useful for just a tiny proportion of people with CF, we can now give this drug at the age of 4 weeks. So we see a baby, we give the diagnosis, and next week your baby is going to be able to start that drug. It's a very special moment when it's a baby like that that comes into our wards. At the moment, we can give that triple that I've shown you the data from the age of 2 years, and trials have just completed between 1 and 2 years of age, which will get put to regulatory, and hopefully there will be a license extension on that. We mustn't forget these people at the top who don't produce any protein. So what's changed? I've described this improvement, and the amount of things that have changed is enormous. We're pretty much rewriting the rule books and the textbooks when it comes to cystic fibrosis now. I'm going to pick out 3 areas of this to talk to you about related to my research programs, but there is an enormous amount of change. And anybody who's here, anybody who's got anything to do with Cystic Fibrosis Trust will know there's numerous different areas of these that we need to be focusing and working on. I'm going to talk a little bit about how do we monitor people's lung health when they are on these drugs and they may be feeling much better and their conventional lung function tests might look like they're much better as well. These are a data set from a long-term follow-up study. I think we go out to something, can anyone read that? Maybe week 96 or something. It's about 2.5 or 3 years of data after people have commenced on these drugs and then followed out over a long period of time. And you can see that obviously there's some wobble with lung function, but it's a pretty straight line. Looks very, very encouraging. Sweat chloride stays down, as you would imagine. This is a more sensitive measure of lung function. Lung clearance index also looking very, very nice and flat, and respiratory symptoms are great. So this looks fabulous. You know, people are looking really, really stable over long periods of time after starting these drugs. I would just raise the question is, are we looking hard enough at some of these patients? And maybe we need better tools than standard lung function tests that we get in clinic. And I'm just going to talk to you about one of these. This is a nitrogen washout test. It's based on multi-breath washout, and you can see at the bottom a nitrogen trace where this person is breathing in 100% oxygen. With each breath, less and less nitrogen is coming out until that's washed out to a predefined level. That predefined level is here in this pink bar. And we measure how many turnovers of lung volume it takes before you wash that out. You could imagine how somebody with really clear, healthy lungs will wash out quickly. Somebody with lots of obstruction and mucus and patchy disease will take longer to wash that out. So this generates a figure called the lung clearance index. Normal is around 7, and it gets higher and higher as you have more disease. We've fairly recently optimized this to do this maneuver at the end. This is a deliberate maneuver to try and free up what we call trapped gas. And that means as we breathe just normally, quietly in the clinic, there are areas of our lungs that might not ventilate because of mucus plugging. If we then do a great big inspiration and an expiration, we can release that and we can find some hidden signal behind that plugging. And this is something that was generated by Chris Short, who is a PhD student with me. This is a person who's done this maneuver and doesn't release loads of nitrogen above that green line. So he's got a normal LCI, and then this new derived system that we come out is the same. It doesn't change. And then you can see this person who's got exactly the same LCI, but this enormous nitrogen, lots and lots of trapped gas, and a completely different value for the new LCI. This is something we think that's quite important and that we should be thinking a little more about. And we've done a project recently where we've combined this with functional magnetic resonance imaging of the lung. Most of you will know MRI is not brilliant for air-filled organs like the lungs, but you can use oxygen to actually map out whether those lungs are ventilating in a nice uniform way or whether they're very, very patchy. And over on the left, there's a healthy person. This is quite a mildly affected CF person with normal lung function. And this is somebody whose lung function is really not bad for an adult, but does have some disease. And if we look at the light blue is normal, the dark blue is less normal. And you can see here from these histograms that the more severe the patient is, the more those histograms lean over to the left. And we've used some of these to map out interventions. How sensitive are these compared with normal lung function? This is just a really small group of children we managed to capture who were starting on that triple modulator drug. Too small, really, to be able to see significant differences with our conventional outcome measures. You can see it's a little bit noisy. But if we look at those same children, either with the physiology test in the middle or with the imaging test on the far right, you can see that quite easily you could have said on a much smaller group of patients whether a drug was working or not. And this is why we think this might be useful, is we could possibly do trials which are smaller in number, bother less people, and therefore cost an awful lot less if we were using some of these more sensitive outcome measures. This is a data set that I think is really a little bit worrying and really important. We've just had this accepted in thorax. And this is looking at the long-term period when that data set I showed you where everything looked flat after they'd started on the drug. We've now had them on the drug for a minimum of 6 months, but actually most of them it was more, and followed out to 18 months. And if you look at FEV1 or conventional lung clearance index, statistically that's not changing. I know it looks like a bit of a slope, but it's not changing. If you look at our advanced physiology, you see that it actually is getting worse. And if you look at it on the imaging, it's getting quite a lot worse. So I think this is a bit concerning. I think we need to properly look at some of these tests and what they're telling us. And possibly we've got sort of rose-tinted spectacles when we're using our conventional tests, and we need to be thinking a bit more carefully. And particularly maybe we need to be thinking a bit more carefully as totally understandably, people who are feeling a lot better want to stop taking their conventional burdensome medicines. They're taking nebulizers, they're taking inhalers, etc., and they're feeling better, they want to stop that. But I think we need to provide the evidence to say that is as safe as we think it is. So that's one of the things that's changed a lot. The next thing that's changed is this. We used to find it very easy to tell people what sort of bugs they had in their lungs because they would come in, they'd cough up a phlegm sample, we'd send it to the lab and we'd culture it. Most people, even those with quite advanced lung disease, find that their lungs dry up when they're on these treatments. And so we have now a bit of a problem in saying to them what actual infections they've got. This is just some data that was collected from our centre, looking at the number of sputum samples or cough swab samples, which is a bit of a surrogate, people put into the lab over a period of a year. And you can see here what happened in the year -1 starting these drugs. That was the COVID pandemic. So we had about a third of a decrease in terms of the number of samples compared with the previous 4 years. But look at year +1 and +2. These people are now on these drugs and the number of samples has really, really plummeted. So it's very difficult for us to be able to say what is growing. This is the priority setting partnership of the James Lind Alliance that was conducted by CF Trust and a number of clinical colleagues. And this is priority number 2. What is the best way to diagnose lung infection when there's no sputum? People recognise that this is an issue. They're pleased not to be coughing up the phlegm, but it does mean that we're a little bit in the dark. So this is the focus of one of the innovation hubs, which is funded through LifeArc and the Cystic Fibrosis Trust. Precision CF is the one that is co-led by myself and Darius Armstrong-James at Imperial. And we've got the 3 others that some of you may have heard a little bit about this morning as well. So just to tell you what we're doing in precision is we're focusing very much on these people who are used to being able to cough up phlegm and now really, really struggle. They love it, but it does mean that they don't know and we don't know whether treatments they're on are necessary or whether they need new treatments. The main aims of this is to develop accurate and acceptable alternatives to conventional culture, and I'll show you a number of those, but also to step back a little bit and recognize the fact that people with a particular type of germ in their lungs don't just have that usually. They usually have a mix of different things. Some of them we call commensals. We might think that they're harmless. We don't necessarily know that they're harmless. And we need to understand a little bit how those organisms interact with each other and interact with the host. So this is the structure of a 5-year program. Workstream 1 and Workstream 2 are quite basic science based in the laboratory. We're looking at the common microbes that people with cystic fibrosis have. We're also looking at some of these commensals. We're mixing them up together. We're looking at the way that they cross-talk to each other. In Workstream 2, we're taking those into slightly more advanced model systems, looking at those mixes of organisms, perhaps on top of epithelial cell cultures, and we're triaging then the most interesting of those into a cystic fibrosis rat model, which is supported by the CF Foundation at the University of Alabama. Workstream 3 is the big clinical one. We're just, I hope, about to launch this. We're calling it the Precision Partners Platform. This is a platform of up to 500 people with cystic fibrosis recruited across the country, all different stages of disease, all different ages. And we're going to ask them to do as many as they wish of a huge number of different sampling types. This is a very sample agnostic study, and it's a very assay agnostic study. So we've got lots of different types of samples, including the gold standard, obviously, sputum, but including, for example, urine signatures, skin and salivary signatures. And then we'll put these through a big barrage of tests, many of them quite advanced omics types approaches. But if you've got BDIs, you can see down here, we've also got my favorite project, which is with the sniffer dogs. Medical detection dogs have been trained to be able to smell Pseudomonas in sputum. And what we're trying to do is to see whether we can match up their ability to smell other diseases on skin by saying, can they also measure these infections on skin? Just to tell you why we're so focused on the polymicrobial environment, it really does matter, this. This is some data from a PhD done by Dominic Hughes when he was a pre-trainee in our group, looking at combinations of Aspergillus and Pseudomonas. And this is an agar plate model where you can see inhibition of the black Aspergillus by the Pseudomonas spots which are growing on there. There's a little halo around them. We also get inhibition by what is produced into the culture supernatant from the bacteria. They also inhibit the fungus. And what I hope you'll see here is time-lapse microscopy. On the left is Aspergillus fungus growing on its own, and on the right is growing in the presence of Pseudomonas. And I think you can see there's a really, really big difference in the way it hyphenates and it grows out. Why is this important? The reason it's important is we did a big registry study and we found that people with cystic fibrosis who had Pseudomonas in their sputum, less frequently we could find the Aspergillus. We know it's there because we can see it molecularly, but it doesn't grow. And so there's what we're calling an invisible threat of the fact that that organism is there. We can't see it because something else is masking it. And we know that sometimes having those two things together is detrimental for people. So it's really important that we know about it. This is still important in the modulator era. I often get asked this, do we still have a problem? Do we still have a job? Some people say, these are probably the best data set to come out of following people through starting this triple highly effective modulator. You can see Staph aureus on the top, Pseudomonas on the bottom. It is an infrequent occurrence that somebody clears those organisms once they've been present for a period of time. The colony-forming unit count may come down a bit, and of course that's a good thing, but it's not transformational. Those bacteria are still there, and my strong belief is that we still have an issue. We still need to be able to treat these. We need more and we need better treatments. And what we're not getting is huge numbers of new antibiotics through any sort of pipeline. And this is looking way beyond cystic fibrosis. This just in general, these are WHO 2019 data. The pipeline is really dwindling of new molecules. So one of the approaches we're taking is to try and make better use of the molecules that we've got at the moment. We're doing this in a project which is funded through LifeArc, repurposing a drug which is used for multiple sclerosis, which I know sounds really wacky and weird, but it was discovered by some collaborators in Denmark that this particular drug, which is called glutamate acetate, acts a little bit like antimicrobial peptides, and it damages the surface and the cell components of bacteria. You can see here some data from my lab showing a real increase in the dye indicator that says these bacteria are damaged. And what we can see is that we can use less tobramycin to try and kill those bugs in the presence of that GA than we would without it. So we've got a group working on this project at the moment. We're finding not only activity against Pseudomonas, but interestingly activity against a lot of different types of organisms that we see in the CF lung. And particularly, it seems to potentiate the activity of aminoglycosides, which is the class that tobramycin is in. So the other thing that's changed enormously is people are leading completely different lives than they have done in the recent past. They're living longer. These are data from the US registry and then modeled data from the UK registry to show that you can see very, very much higher life expectancies than has been seen in the past. This does mean that we now have to cope with an aging population who may get all sorts of different diseases than we've seen in the past. And just to end off on a high, I'm going to tell you about one of those projects, which is about pregnancy in people with CF and women with CF. We've seen a threefold increase in the numbers of pregnancies since modulators have come along. This is both because of the biology of CFTR in the cervix, but also I think it's the difference in optimism and hope in terms of their lives. And this is another CF Trust funded SRC, which is led by my colleague, Dr. Imogen Felton, and is looking across the board at women with cystic fibrosis who are getting pregnant, looking at optimizing their health, but also looking at being secure and safe in the knowledge that those fetuses which are exposed to these drugs that we know not very much about are actually going to be as safe as we think. There's a couple of references here that you can see, including a Delphi consensus that we've recently put through the European CF about how to monitor these babies. So in terms of take-homes, yeah, CF has changed enormously for many people, for most people, I think, but we do need to remember those people that can't get access to modulators. The rule books need rewriting. They're very different than they used to be. We've got new challenges now, and I think it's really important that people don't leave this room thinking we've cured cystic fibrosis. We've got a lot more to do. I think it's important that we're realistic and not overly optimistic. Otherwise, people will begin to feel very let down as time goes on. And so the message really is there's an awful lot more to do. Thank you very, very much to LifeArc and CF Trust for their ongoing funding and support, and thank you all for listening.